So it's my pleasure to introduce today's seminar speaker, Mandy Gibson. She is, I'll go chronologically. So she got her bachelor's degree from Amherst, then she did her PhD at Indiana working with Kurt Lively. She had an NIH first postdoc at Emory, and also did some teaching at Spelman during that time. And then she's been at the University of Virginia since 2019, which strikes me as a particularly interesting time to have started a faculty position. She has published lots of really amazing papers and won lots of awards. To just subset the awards, they include the Ruth Patrick Award for a Best Poster from the Society for the Study of Evolution, and has 2 more awards just from that society. So the Thomas Henry Huxley Education Award and the Dobzhansky Prize, which is really like quite a major honor from them. And she's also received the John Maynard Smith Prize from ESEB. She's currently supported by NIH, which I find totally amazing because I find NIH like totally incomprehensible. And so today she's going to tell us about some really exciting work she's been doing with that support.
Okay, is it working? Can you hear me in the back? Awesome. Okay, well, thank you so much for the introduction, and thank you all for having me. It's been so nice meeting with so many of you today. So my lab studies the ecological and evolutionary dynamics of antagonistic interactions. We work on a lot of different host-parasite systems in my lab, probably too many. I'm just going to tell you about 2 today, and these are going to be stories about parasites imposing selection and acting as architects of host populations. Specifically, we'll be looking at parasites imposing selection on sex and dispersal. So these are traits that are fundamental in determining the amount, nature, and distribution of genetic variation in host populations. And I got excited about these topics in particular because of a paper that I read early in my PhD. It's a paper written in 1978 by John Janicki. It's one of the original papers putting forward the idea of the Red Queen hypothesis, the idea that parasites can maintain sexual reproduction in their host populations. So I'll snapshot the key bit that's inspired me for a long time. Janicki wrote, most organisms are probably under intense pressure to escape in one way or another the biotic component of the environment. He's being a little vague, but you can tell from the paper he's thinking about parasitic lifestyles when he says biotic component. 2 escape routes are open. One is escape in space, that's dispersal, and the other is escape in time. Right, so that's sexual reproduction and the production of offspring that have new or rare genotypes that can escape local parasites. Okay, so there's a big idea here, right? Janicki's arguing that parasites are imposing selection on these 2 really fundamental host traits, sex and dispersal. Right, and these are also both traits that have really confounded evolutionary biologists for a long time. Right, so I was immediately captivated by these ideas. Right, and increasingly my lab is really looking at sex and dispersal as 2 sides of the same coin, right, as these alternate solutions to the same problem. So that's what we'll talk about today. I'd like to start with sex. So the ubiquity of sexual reproduction is one of the core anomalies in evolutionary biology. There's also some definitional problems, so I'm just going to start out with those so we're all on the same page. And I would like to recognize Project Biodiversify that gave me some of the language, right, to be able to talk precisely about these problems. Right, so when I talk about sex, I'm talking about sexual outcrossing. This is when 2 biological parents come together and have the potential to produce offspring that are genetically distinct from their parents and from one another, from their siblings. We're going to contrast that with asexual reproduction or cloning. That's one biological parent produces an offspring that's genetically identical to it. In the first part of the talk, we're going to meet systems that are dioecious, so they have males and females. And all I mean is the males have the small gametes, the sperm, and the females have the large gametes, the eggs. When we start talking about nematodes, we'll be talking about some hermaphrodites. And these are individuals that have both gametes, sperm and egg, and they can self-fertilize. Okay, so those are our definitional problems. Sex is also an evolutionary problem, and that's because we think it's costly. Specifically, we think the production of males is costly. I think this is nicely shown by this simulation by Kurt Lively. We have time on the x, population size on the y, and we're imagining we have a sexual population at equilibrium. So meaning on average, every female is going to mate with a male, and she'll produce just enough offspring to replace them, one male offspring, one female offspring. Okay. Then we introduce an asexual mutant. She's just like her sexual counterpart. She also makes 2 offspring, but because she's cloning, these are females, right? So she's putting 100% of her resources into daughters that can now bear more offspring, whereas this sexual mom has put 50% of her resources into a son that can't directly bear offspring. Okay. So that results in a 2-fold higher per capita birth rate for our asexual lineage. We call it the 2-fold cost of males. And the result is that sex is outcompeted, right? It goes extinct in this simulation, less than 10 generations, which is very quickly. So, of course, that's not what we see. Sex is alive and well, right? If we look outside and we just look at the animals, fewer than 1% of animal taxa adopt this seemingly more efficient strategy of being obligately asexual. And if we zoom into the nematodes, which I like to do, because in this case, reproductive mode can evolve a little more readily than in many other animals, even in that, the nematode phylum, we see that fewer than 30% of nematode taxa adopt this strategy, right, of being selfing or asexual and making few to no males, right? Being outcrossing, making lots of males is far and away the dominant strategy. Okay. So this conflict between theoretical predictions and what we actually see in nature has caused a lot of consternation in evolutionary biology, nicely captured here by G.C. Williams in 1975, where he wrote, "There is a kind of crisis at hand in evolutionary biology." He's talking about sex. Now, so one of the hypotheses proposed to resolve this problem, and really only one of 2 hypotheses about this that stood the test of time, is the Red Queen hypothesis. And this is the idea that co-evolving parasites target common asexual lineages, and in doing so, they counterbalance the cost of sex and can maintain sexual females in the population. Okay, we can see that if we look at this simulation. So here, sex went extinct under the 2-fold cost of males. We add in co-evolving parasites, and we see sex maintained. And we can figure out why if we look at the frequency of infection of our asexual in blue and our sexual in red lineages. So what's happening is our asexual lineage increases in frequency, and as it does so, the local parasites lock onto it, right? They adapt to infect it. It becomes incredibly infected, more infected than the population mean, which drives our asexuals down in frequency under the weight of that parasite selection. Now they're rare, right? And they're flying under the radar of local parasites, right? So they're under-infected or less infected than the population mean. Meanwhile, our sexuals are kind of bopping along, right, at a relatively low infection frequency. The idea being that sexual moms are continually making offspring that have rare genotypes that evade the local parasites. Okay, I like presenting the Red Queen this way. It contrasts with my original, I think, simplistic understanding of it, which is just that asexuals get hit harder by parasites than sexuals. This is really saying it's not about the mean, it's about the variance, right? So parasites are imposing a lot more variance in infection frequency and thus in fitness in our asexuals than our sexuals, and that's what's maintaining sex in the population. Of course, that kind of maybe more nuanced perspective is a lot harder to test. So I wanted to set out to do this. How do we do it? Well, ideally, we have a natural system that we can track through time, and this little snail, Podomopyrgus antipodarum, has proven incredibly valuable in that respect. Because there are a few things about this snail that make it helpful for these questions. So it has obligately sexual males and females that coexist with obligately asexual females in the lakes and streams of New Zealand where we find this snail. So we have them coexisting and competing with one another in a single population. Sexuals are diploid, asexuals are triploids. We can tell them apart pretty easily. And the assumptions of the problem are met in this system. So there is a 2-fold cost of males. I was able to figure out by mixing asexual and sexual females together in the absence of parasites, the asexual females increase in frequency over the course of a single generation. And the magnitude of that increase is what we would expect, what we would theoretically predict if there was a 2-fold cost of males. And yet males and sex, they're doing fine, right? They are all over New Zealand. So the skeleton of the problem is there. And of course, there's parasites. Okay, so this is a healthy snail. Well, it was healthy until it was pulled out of its shell by Gabe Harp. I didn't do that. And this is an infected snail. So much of its tissue has been replaced by the metacercaria or the cysts of this trematode. Atrophalophorus winterbornii. In particular, the gonads have been replaced by parasites, so it's sterilized. This is really strong selection. And last but not least, we have really beautiful field sites. This is Lake Alexandrina. I spent a lot of time here. It's on the South Island of New Zealand. It's a wonderful place to be, and you can just walk up to the shore. You can sieve snails right off the vegetation on the shore. You can, if you are Kurt, my mentor, you can swim out further and you can sieve them off the vegetation in the deeper waters. And then we just package them up on the shore. So that's what my collaborator Linda Delph is doing. You can bring them back to the lab, figure out if they're male or female, if they're parasitized, and if they're female, you can go in and say, okay, are you diploid or triploid? Are you sexual or asexual? Okay. So I wanted to test this model of the red queen by tracking asexual frequency and infection through time. And I was able to do this because we had a historical data set from 2001 to 2005 that was originally collected by Daniela Vergara, and then a new data set that I collected from 2012 to 2016. So that's 10 years of field data spanning 16 years. So we'll look at asexual frequency and infection in that first period of time. So in 2001 to 2005, asexuals are pretty common, and they are really infected, at least in 4 to 5 years. So I'll just point out, on average, more than 50% of females are infected, asexual females, and that means that more than 50% on average of females are sterilized by this trematode, by this single species of trematode in this lake. Okay, we come back in 2012, and things have really changed. Asexuals are pretty uncommon, and actually in 2012, in half of my sites, so 3 of my 6 sites, I found no asexuals. And they're really uninfected, right? They're much less infected than the sexuals, much less infected than the population mean. Right, so this is consistent with this movement from being common and over-infected to being rare and under-infected. And it's driven by the asexual. So if you look, the sexuals are reasonably constant. It's the asexuals that are fluctuating, which actually that generated, I think, my favorite result of this whole study, which is that if I look at all females across all 10 years of data on all sites, and I look at the frequency of infection in sexual and asexual females, there's no mean difference in infection between sexuals and asexuals. What's different is the variance, right? Asexuals are oscillating from no infection to really high infection through time. Okay. So as I was doing these field collections, I was also running sort of 4 independent years of these experiments in these outdoor mesocosms, where I was taking snails and parasites from the natural population and putting them into these mesocosms. And I'm not going to show you the data for that, but I'll just tell you a couple take-homes. So first, I was able to figure out that these asexuals, they're not just really uninfected because they're somehow like avoiding infection in the field. When we take them into the lab and we expose sexual and asexual females in these mesocosms to a controlled dose, we force them to encounter the parasite, they are much more resistant. So that tells us that local parasites are doing a relatively bad job of infecting these rare asexuals, right, which is consistent with the red queen model. The other thing we determined is that asexuals have a massive fitness advantage. So in the absence of parasites, they're increasing in frequency. So this was over the course of a year. We would track the change in frequency in these mesocosms. They would increase in the absence of parasites because they're not paying the twofold cost of males. When you add in parasites, they do even better, right? Because the sexuals are getting more infected than the asexuals. Okay, so that gave me somewhat of an unusual opportunity to falsify the red queen in this natural population. We said, okay, the red queen at this stage is predicting that asexuals should be increasing in frequency, right? Because they're not paying the twofold cost of males, and they're currently not getting hit by parasites. If they fail to increase in frequency, that tells us that something is more powerful than parasites is preventing the spread of asexuals in the population. And while that wouldn't reject the red queen, it would at least reject it as the primary explanation for the frequency of asexuals in these populations. Okay, so I went into my field data and I looked at the frequency of asexual females from 2012 to 2017, and Zoe Dingus actually collected this last data point. She was a graduate student at Indiana University. And what we see is a massive increase in asexual frequency. There's a 4-fold increase through time. Okay, so this is what we predicted. We can't reject the red queen just yet in this field population. But we can ask some questions, like, what is driving this increase, right? Is this from a spread of one or a few common clones? We think so. There's some data from recent data from Yuka lab and Ayavog suggesting that might be the case, but we don't know for sure. And more importantly, when are parasites going to adapt to these, we think, common clones, right? We certainly think they should have done so by now. That we don't have an answer to, but it's been about 5 or 6 years, so this is a great time to come back to this system and see the extent to which it's, if and how it's turned over, right? Are asexuals now getting hit by parasites? Okay. So I've just shown you some evidence consistent with the idea that co-evolving parasites drive variation in the relative fitness and frequency of asexuals. We think it's those oscillations that can maintain sexual females in the population in the face of the twofold cost of males. So this work and much other work in this system by myself and others has now continually sort of supported the idea that indeed sex may function as escape in time from parasites. Okay. And now I'd like to move on to the second part of the talk. So I'll remind you about this quote that so inspired me. So this paper, it really goes on to talk about the escape in time hypothesis. This paper is about the Red Queen hypothesis. That's what it's known for. And after its publication, the Red Queen hypothesis has taken off, right? It's now one of the best known and most extensively studied hypotheses we have in evolutionary biology. Meanwhile, the escape in space hypothesis, the idea that dispersal is a strategy to avoid parasites, has remained in the background, right? It's received very little empirical and theoretical development. And I think that's a problem. First, dispersal, just like sex, is an important evolutionary problem. I'll talk about that more in a minute. And by not exploring parasites as a driver of its evolution, we might be missing a potentially really powerful and omnipresent factor that explains why and where we see it. Secondly, in the disease world, we typically think of dispersal as a fixed parameter, right? But if we allow it to vary, evolve, or even evolve in response to parasites, it really changes how we think about the evolution of other traits like sex that are evolved in defense. Right? So, and that's what Janicki's kind of alluding to here, right? He's saying that sex and dispersal, not only are they strategies, but they might be alternative strategies, right? You might do one or the other. Okay. So this gap is really what's motivating the second part of the talk. And this is new. Some of the data you're going to see is really pretty uncooked, like the past maybe few weeks. So I'm eager to hear feedback and to hear what you think. Okay. So just like the evolution of sex, dispersal is a problem. It also has some terminology problems, so we'll start there just like we did. So I like this definition by Ophélie Rance. It's any movement of individuals or propagules with potential consequences for gene flow across space. I like this definition because it puts the why should we care in the definition, right? And indeed, gene flow is one of the primary determinants, dispersal is one of the primary determinants of gene flow across space. And it's critical to the persistence of species in the face of local extinction. I'll mainly be talking about natal dispersal. So this is dispersal from your birth site or origin site to your breeding site. This is really a question about when and where should I reproduce. And we can break dispersal down into 3 parts that you'll hear me touch on sort of each of these. There's the departure stage, right? How likely am I to leave? There's a transfer stage, which is sort of out in the world, moving to the next, making your way to a new site. And then there's this colonization or settlement stage where you've landed in the new site. Okay, and you can immediately see from this diagram why dispersal has been an empirical challenge, right? It has multiple moving parts. Each of them is complex, and they're really hard to track out in the wild. Okay. Dispersal is also an evolutionary problem, and so just like sex, it's because we think it's immensely costly, right? And you can sort of think about the costs that are realized at each of these steps. So first there's departure, right? You're giving up reproductive opportunities in a known site, a known quantity. You're going out into the world, you're assuming all these risks in the transfer stage. And then you land in this new site that you may or may not be suited to, right? That may or may not be better than where you came from. Okay, and indeed in this really canonical paper by Hamilton and May, they describe these almost suicidal ventures that organisms undertake as they're dispersing. And yet you would be hard pressed, right, to find an organism that doesn't have dispersal as some part of its life history, right? From seed dispersal to these sort of short distance dispersal between breeding colonies to, of course, these really charismatic migration events. So just like sex, this conflict between the costs and how common dispersal is has generated a lot of theoretical attention, trying to understand what drives the evolution of dispersal, what maintains all the variation in dispersal behavior we see out in the wild. I mean, one of the clear take-homes of that theory is that dispersal can evolve as an adaptation to ephemeral habitats, right? Specifically, we know that, or quite interestingly, we know that predictable habitat decline can favor individuals that disperse in response to ecological cues associated with habitat decline. Okay. So the theory has the advantage of being able to be somewhat vague about what we mean by habitat decline, right? You can have a parameter that's sort of like, this is how likely this habitat is to go extinct, but as empiricists, we want to pin that to something, right? We want to know what are the actual factors that are associated with habitat decline and are imposing selection on dispersal. And I think this is where parasites fit in, right? So parasites, they're really common, they impose really strong selection, and at least in theory, right, they increase over time, over the course of an epidemic, right? They do so predictably, right? So they could be really general, unreliable harbingers of habitat decline. So this idea has been around for a long time, and indeed, parasites have been proposed to explain the evolution of seed dispersal, the evolution of this early life decision to sort of stay home or leave, and the maintenance of long-distance migration. And while there's been fairly limited empirical tests of this idea, well, there's one thing that we do see fairly consistently, and that is that parasites do indeed seem to trigger dispersal, so either increase dispersal propensity or distance, right? We see that in cliff swallows here in this fundamental paper from 1992. Okay, so we see this repeatedly. It seems to be a real phenomenon. At the same time, the other thing we see repeatedly is that parasites increase the cost of dispersal, right? So infected monarchs are less likely to complete their migration than healthy monarchs. And I think more interestingly, like these two papers are examples of systems where you see the two at the same time, right? You see parasites cueing dispersal while simultaneously reducing the ability of hosts to complete the dispersal, which leaves us in this kind of uncomfortable place, right? And it leaves open this key question, right? Is dispersal really an adaptation to avoid parasites? Or, do these benefits of avoiding parasites actually counterbalance this risk of dispersing while infected? Or are these stories of parasites triggering dispersal actually just maybe byproducts or potentially manipulation by parasites? Okay. So we were interested in solving this problem or addressing this question, and we're doing so using the nematode, C. elegans, the model nematode. You might be familiar with C. elegans. It's used as a model for many things, usually motivated by its experimental and genetic tractability. All those things are true. I'd like to argue that it's its ecology that motivates it as a model system for studying the evolution of dispersal. Okay, so I'll tell you a bit about what we know about it. If you know C. elegans, this is part of the life cycle you probably know. I'm going to call this the within patch life cycle. What this looks like in the field is you have patches of rotting vegetation where you have bacterial blooms. A few nematodes are colonizing those bacterial blooms. The estimate we have right now is like 3 to 10. That will probably change over time as we get more information. But C. elegans is typically selfing, so most of the time it seems like it's a single selfing lineage that's colonizing a given bacterial bloom, but you can see coexistence of multiple selfing lineages. They reproduce really rapidly, so hundreds of babies over the course of a couple days, and this patch gets really, really dense. Right? So babies that are born into this patch are getting cues of high density. They use pheromone to sense their own density, and then also cues of starvation. And that's going to cause them to develop not into reproductive adults who are going to reproduce in the habitat, but as dispersal stage larvae, which you're going to hear me refer to as Dauer. So Dauer is German for enduring, and indeed these are like diapause. They're akin to diapause stages. They're long-lived, they're environmentally resistant, they're non-feeding. And they have these amazing behaviors, like what you see here, where they're waving around on their tails, or maybe not tails, whatever, their ends. And what they're trying to do, it's called nictation. It's really widespread behavior across the nematode phylum. They're trying to attach to an invertebrate, like an isopod or a snail. So they're going to passively disperse to a new bacterial bloom. So these nematodes are living in these really ephemeral metapopulations, right? These patches are blinking in and out across the landscape. So really the persistence of a lineage requires not just that they can survive and reproduce within the patch, they have to pump out lots of dispersers, which then have a high probability of colonizing a new site, right? That's what sort of maintains a lineage in the metapopulation. Okay, so I think that's one thing that makes C. elegans a great model for studying dispersal, right? It's a really, really naturally relevant, important part of their life cycle and their evolution. There's also an enormous amount of genetic variation in dispersal propensity. So specifically, selfing lineages vary hugely in their sensitivity to cues like density. So some make lots and lots of dispersers at low density, and some make none even at high density. Okay. You can also replicate and manipulate the whole process in the lab by treating petri dishes as patches and moving dispersers between those petri dishes. Okay. And finally, there is a parasite. So we study Nematocida parisi, or the nematode killer from Paris. This is the first natural parasite of C. elegans ever to be described in 2008. It only took 40 years for them to find it. But it's actually really common, it's just no one was looking. So we think this is the most common natural parasite of C. elegans. It's a microsporidia, right? So this is a group of obligately parasitic, I'm gonna call them fungi. And they're trophically transmitted. So nematodes are munching bacteria, they bring in spores. The spores infect their intestinal cells, that's what you're seeing here. And then they pass spores back into the gut, which then are shed into the environment with the feces. And those spores can build up in the environment. You can get these massive epidemics, at least in contained petri dishes. Okay. So our aim was to study, to use this interaction to address this question, right? Is dispersal an adaptation to avoid parasites? So on to some data. First, I'd like to convince you these parasites are worth avoiding, right? This is really important in theoretical treatments of this problem, right? The parasite has to be sufficiently virulent to offset all the costs of dispersal that we think exist, right? So are they virulent? And this is work that I'm gonna summarize pretty quickly of 2 undergraduates, Tori and Emily, 2 technicians, Sarah and Anne, and my graduate student, Lewis. And really what they're asking is if we expose, this is looking at the within patch stage. So if we expose young larvae to parasites and we let them reach reproductive maturity, how is their fitness affected, right? Is their survival affected, fecundity, and population growth? I'm just gonna show you fecundity because we think that's the primary effect. So we look at the number of offspring produced through time by hermaphrodites in control and exposed conditions. And we see a massive reduction. This is a 72% reduction. So the thick lines are the means, the thin lines are individuals. This was at pretty high dose. And also we used the standard lab adapted strain. You might hear me call that N2. We subsequently learned that this strain is actually pretty susceptible to our parasites. So we said, I don't know if this result is actually general. So we wanted to kind of expand it out. So we collaborated with Sender, which is the C. elegans natural diversity resource, to get 20 genotypes or 20 strains of C. elegans from Hawaii. We think Hawaii is part of the ancestral range of C. elegans, and we find a lot of genetic variation on these islands. So we picked 20 really highly divergent strains. We exposed them to a couple different doses of our parasite, and then we tracked their daily fecundity through this sort of homemade imaging setup that we have that Sarah and Tori and Anne are working at. So I'm going to show you the number of offspring produced in control and exposed populations. And you're going to see 20 lines, so those are 20 genotypes. It's just the means across each of those strains. Okay, so yes, for the most part, parasites are reducing fecundity. Most of these lines are going pretty nicely downwards, some as much as 60% reduction. There are some nice exceptions, which means we have genetic variation, right? So this strain here is almost totally fine. And a couple of these strains, like this one, actually increase their fecundity. That's almost a 25% increase in egg production in the presence of parasites. So just file that away for a later result that you'll see. Okay, we also were able to determine that this is dose dependent, right? So we took the average across all 20 strains, is what you'll see here is offspring produced in control, high dose, and then this is actually moderate dose. It's not that low. And we can see that you do worse when you're exposed to higher doses of parasites, which is not surprising. Okay, so yeah, these parasites are nasty, they reduce fecundity, and they do so in a dose-dependent manner. So the inference there is that if you wait and you stick around longer in a patch later in an epidemic when there's lots of parasites around, you're going to suffer more fitness consequences. Okay. We then asked whether or not parasites trigger dispersal. We thought this was intuitive, right? If it's an adaptation to avoid parasites, we would think that hosts would be dispersing it in response to those parasites. Okay. So when we're moving here now from the within patch stage to this between patch stage and asking if the presence of parasites can cue the formation of our dowers. Right, and this is work of Lewis and his undergraduate Sarah. Actually, all the data I'm going to show you from now on is work out of Lewis's dissertation that he's doing with Sarah. So what they did is they're measuring the proportion of individuals that are developing as dower or as dispersal stage larvae. They do it with 2 different genotypes. We kind of learned our lesson on not doing things just with N2, which is the more susceptible genotype. This is a divergent genotype that's commonly used when folks want to feel like they're working on a wild genotype. It's fine, it's genetically divergent, it's okay. And they do it in the presence of pheromone, right? So I'll remind you, this is how worms sense their density. So as you increase pheromone on the plate, worms think they're at higher density and they're more likely to disperse, right? And we do this because it's actually quite hard to get dower to form unless the worms are at really high density. We don't have any problem getting the worms to high density, but we don't love conducting experiments at really high density. It's very messy. So we kind of compromise and we trick the worms into thinking they're at high density. Okay, so then the question is, if we add in parasites, does that increase dispersal or the formation of dower larvae? And yes, at least in this first genotype I'm showing you, we see more than a 2-fold increase in the production of dowers, but not in the other genotype at all. Which is exciting, right? There's genetic variation in this response, which we can potentially actually use to get at the genetic basis. It does raise the question of what's actually happening. We think that N2 is the weirdo here. It's kind of the lab-adapted strain. But Lewis and Sarah are expanding this out to a couple more genotypes to actually characterize this response more thoroughly. They also did a follow-up. This is all with this genotype CB4856. And here what they did is they basically did freeze-thaw cycles on the spores, which keeps the spores intact, but makes them non-infectious. And when they repeat this experiment with these non-infectious spores, they see no response. Right, so that gave them a little bit of a sense of like what is driving this response. So it's not just spores in the environment. It's actively infectious spores or potentially signals from infected conspecifics. We're not sure which one. Okay, so these parasites are nasty, they're triggering dispersal, but we also found out that dispersing while infected is really costly. Okay, so similar to what we've seen in some other systems. So we found this out by rearing populations in the presence and absence of parasites and just letting them go like we think they do in the wild, letting them get really dense and form dowers. And then we come in and we simulate dispersal for them. We sort of pretend that we're an isopod. We pick up the dispersers and we move them to a new plate that has plenty of food. So this is where they should do great. They should be able to colonize. And then we just said host dispersal is successful if you're able to reach reproductive maturity and reproduce in this new site. It's not successful if you fail to do so. Okay. So yeah, you might be thinking we've made things really easy for our hosts. We've skipped all the components of dispersal except this colonization phase. I totally agree. I think you'll see that makes our results fairly conservative. Okay, so we have success rate on the Y. So this is the fraction of dispersers that make babies in the new site. It's 100% if they're coming out of healthy populations, right? So 100% of individuals can come out of dower and reproduce. I think that this is one worm out of like 60 that managed to mature and reproduce. Okay, this is a massive cost of dispersal when infected. I was really surprised by this. So I'll remind you, when I showed you sort of those within patch fecundity effects, this parasite's not nice, but it's by no means sterilizing. But when you zoom out to this between patch or dispersal level, suddenly the parasite is becoming sterilizing. So this is the first time I've experienced a result like this where you realize that you would be missing the fitness effects of the parasite if you didn't account for the dispersal stage, right? Or you'd be grossly underestimating the fitness effects of the parasite. I didn't believe this result. We've repeated this experiment now probably far too many times. It has held. I will show you one extra experiment that we did. So this is with N2, that susceptible host genotype. Maybe we're just missing something. We're not. We expanded it out to multiple genotypes. We have N2 and then 3 others. They all show the same thing. Actually, this genotype does even worse. This sort of CB4856 is a little bit more of a resistant genotype, but even then, that's a 60% decline in dispersal success. So we're seeing some variation, but overall, a major cost of dispersing while infected. Okay, so we're finding that, again, just like we've seen in other systems, parasites are triggering dispersal, and yet it's obviously not a good idea to disperse while you're infected. So what do we make of this? The possibility remains that this sort of triggering of dispersal is actually just parasite manipulation. The alternative hypothesis that we're exploring right now is that parasites are selecting for dispersal early in an epidemic when prevalence is low. Right? The idea here being that your risk of infection is low early in an epidemic, you're likely to be entering dispersal when you're not infected yet. And also, we're assuming that low prevalence is a reliable indicator of what is soon to be high prevalence. Okay. This hypothesis comes from one of the few theoretical treatments of the evolution of dispersal in response to parasites. And what they established is that the conditions I just set up for you, right, that parasites are virulent, and then they ratchet up the cost of dispersal, that those conditions can create selection for dispersal early in an epidemic, right? In this case, sort of conditionally in response to cues of low parasite prevalence. Okay. So I'm going to show you a couple bits of data, again, from Lewis and Sarah that I think are starting to support this model. But some of these data you'll see are really, really new, and we're still making sense of them. Okay. So first, Lewis and Sarah are really interested in whether or not dispersing early out of an epidemic, out of an exposed population, actually allows you to avoid this cost of parasitism. So we've got this infection that's growing through time, and we're going to compare sort of how early and late individuals that enter dispersal early versus enter dispersal late do over the course of an epidemic in relation to the control. Okay, and we're just going to look at that that dispersal success, right? Can you reach reproductive maturity and make babies in the new site? So if we look at success rate of just our dowers that are collected late, so individuals that enter dispersal late when the risk of infection is high, we see what we saw before, right? So control are fine, but individuals from exposed populations are doing really poorly. Just one day earlier, I'm not sure why we ran the experiment this way, but we did. We just looked one day earlier. Dowers that have formed one day earlier are totally fine. So we sort of lose that cost of dispersing while infected. Suggesting that by entering dispersal early, they really have managed to avoid parasites in the environment and this cost of dispersing while infected that they would surely incur if they stayed just a little bit longer. Okay. So at this point, we've done all this work that's sort of looking at these snapshots of these populations, right? But these are really dynamic populations, right? They're growing in size through time. The risk of infection, the prevalence of infection is growing, and we think the production of dispersers is increasing through time, right? And so finally, Lewis and Sarah are like, we're sick of the snapshots. We're just embracing it, and we're going to run all these populations through time and sort of break them down regularly to really figure out what's going on. So they had multiple populations exposed and controlled through time, breaking them down every day. And they were interested in several things, but ultimately they were interested in the number of reproductive dispersers that are being produced out of these populations, right? So the idea here is that as populations grow in size, they're pumping out more dispersers, simply just by numbers and density. But that as the risk of infection grows, most of those dispersers that are being produced in infected populations are becoming inviable, right? So you get this peak of reproductive dowers that then collapses, right? Because most of them have now entered with this really high risk of infection. They're very unlikely to be successful. But we don't expect that in our control populations, right? We just expect the number of dispersers to increase as the population grows in size, and they're not suffering any fitness consequences. So we have these two really different peaks, and this should impose really strong selection in exposed populations for individuals that are entering dispersal early. So I'll just say in advance, the results broadly support the model, I think. They don't do so in the way that I expected, which you'll see in a second. So the populations were started out here, I think at about day -4, and this is just to kind of get the host populations going a little bit. And then we introduce a really low dose of parasites at day 0 after that burn-in, right? And that's to simulate sort of just the very earliest stages of an epidemic. We're trying to do our best to simulate what we think is actually happening. And then they started tracking population size through time. And immediately saw something that we really didn't expect based upon the fact that we know that these parasites reduce fecundity. That exposed population is orders of magnitude larger than the control populations. I'm happy to talk about why we think this might be happening. For the moment, I'll just say it really is happening. We've repeated this now with multiple replicates and blocks. Lewis has convinced me this is real. And it actually has the expected effect, right? So if we look at the number of dowers, the dispersers that are being produced by these populations, again, it's orders of magnitude higher in exposed than control populations, right? Because these are much more dense, right? We do think that this is driven by density. So I know I showed you earlier that the parasites can cue dower formation. We actually use the genotype here that doesn't do that, right? It's not able to sort of increase dower formation in response to parasites. We kind of wanted to simplify things. So we think that this is purely a response to the increased density of exposed populations. Okay, but the parasite's spreading, right? So I'm not going to show you prevalence data because Lewis wants to get a few more replicates, but or analyze a few more replicates from these populations. But I will tell you that basically parasites they're like pretty low until about this day, and then they shoot up. And that's the first day that we think there's a big round of new transmission that's happening. All right, so accordingly, the success rates, the fitness of our dowers drops from about 100% to down to 25%. So about 75% of the dowers that are coming out of these, coming out of this later epidemic are unable to reproduce. Right, so then we can adjust this graph from number of dowers that are produced to the actual number of dowers that are reproductive, right? And what that does, that's 100% of control individuals, but it's gonna knock our exposed production down. This is a log scale, so that's a larger drop than it might look like. So now suddenly what we're seeing, right, is that there's a peak in the production of reproductive dowers in our exposed population. There's this window where exposed populations can produce dispersers that are gonna be successful at dispersing. And that peak is days earlier than in our controls. Our controls actually, we just stopped at 10 days. They might keep going up. But let's say that's their peak. This is 5 or 6 days. The peak in exposed is 5 or 6 days earlier than what we see in control, this is an organism with a generation time of 3 to 4 days. So that's a massive shift in sort of the phenology of dispersal in this system. And it broadly supports our model that we think that we should see selection for early dispersal in the presence of parasites. Okay. So we pose this question, is dispersal an adaptation to avoid parasites? We're particularly interested in this hypothesis that parasites are selecting for individuals that are dispersing early in an epidemic when risk is low. I've showed you some hints. I was going to skip ahead to my future directions, but I'll wrap it up a little bit more. Right, so what we're seeing is that hosts indeed are dispersing in response to parasites, but dispersing while infected ends poorly. And so when we put these things together in this more dynamic experiment, we're seeing intersection of those two in a way that indeed supports the hypothesis that there's strong selection to disperse early or in the early stages of an epidemic. We would like to directly test this with experimental evolution. So we have a lot of genetic variation and dispersal propensity in this host. So we can take all that genetic variation, allow the host populations to evolve in the presence and absence of parasites, and we expect dispersal behavior to evolve, specifically to see individuals that are dispersing much earlier in the presence of parasites. Either like constitutively, they just do disperse early, or possibly in risk, like sort of increased sensitivity to cues of density or parasites. We're also really interested in its consequences for other defenses, right? So if dispersal is really allowing hosts to avoid contact with parasites, it should have consequences for the evolution of other defenses like resistance that operate after you make contact with parasites, right? After avoidance has broken down. right? So allowing dispersal to vary and potentially evolve really does change our predictions for how we think these other defenses should evolve. Specifically, we expect a negative relationship. So we can test this with trait correlations and experimental evolution. And lastly, I've kind of ignored the fact that dispersal and transmission is happening in metapopulations, right? I did this on purpose, right? I think it's really hard to understand the complex dynamics of a metapopulation if you don't know what's happening within a single patch. And I think you've probably seen that even within a single patch, it's pretty complicated. But we can't ignore it, right? Because dispersal is not advantageous if individuals are dispersing to nearby patches those patches need to be, on average, have lower parasite prevalence than the patch they came from, right? So if in general you're dispersing to patches that are just as sick as the one that you were just in, that's not going to favor dispersal. Right, so we're planning some field collections in orchards where we know we can find C. elegans and these parasites living on fallen apples. Okay, so I'll end with that. I've told you 2 stories now about these traits that we think function as parasite escape mechanisms, sex as escape in time, dispersal as escape in space. And I would particularly like to thank my lab, especially Louis and Sarah, who generated a lot of the data you saw at the end, as well as my funding, and several folks that have supported this work. And thank you for listening.
Maybe I'll just get us started with questions. Thank you for that talk, and I'm curious about those genotypes that you think maybe had higher fitness in the presence of the parasites, and are they eating them, or what do you think's going on with those?
I don't know. So that is something. So my postdoc Linyao and my tech Tessa are really pushing forward on a project right now. I think I have. Well, maybe I'll just go back. I have a slide somewhere, but yeah, so we don't, basically what they're doing is they're pairing this. So this is just offspring production. We don't really know what's going on under the hood. And so they're currently going in, doing exact same exposures and then asking how infected are all of these? So these ones that do better either don't have a fitness reduction or actually increase. Are they just really resistant? They're not getting infected, they have low prevalence, they have low load. Or actually, the hypothesis I favor is that these are just sort of terminally investing, right? They have potentially massive infections and they are just dumping. They're just I'm just gonna get these babies out. So that's what I think. So we're trying to essentially say, right, what's your load and how does that correlate with the fitness effect? My suspicion is it does not correlate at all. That these are, I think these are probably not just resistant. I think, but what actually strategy they're using, I don't know. Yeah, that's a great question. But I think there's some dose dependence, so I'm not showing this, but for each of these genotypes, we have how well they do at low and high dose. And in most cases, we see this uptick in reproduction when we expose them to a low dose, but when we shift to a high dose, it goes away. So there's some dose dependence, and perhaps these genotypes are just sort of some way more resistant or tolerant, and we're tapping into that when we expose them at low dose. But I think maybe all of these would do that if we ratcheted to really low dose. Yeah.
Can you check it toward that? Maybe Luis, then Marvin.
Thanks. Thank you for your talk. I am very interested in how the concept of dispersal is an adaptive strategy to avoid parasites, but I'm also thinking of so you can also see some nematodes having developed defense strategies, defense mechanisms chemically or bodily structural defense strategy against parasites.
Sure.
And I'm kind of thinking of how do you do host balance between the costs for dispersal strategies or this defensive mechanism strategies, and does this trajectory is dependent on the fitness landscape existing like evolutionary context of the nematodes. Like,
Yeah, so you're, to rephrase what I think you're asking, like, are you sort of interested in this interplay between dispersal as a defense, this sort of avoidance defense, versus, immunological or other physiological defenses that you might use to combat parasites?
yeah.
Yeah, we're really interested in that. Totally. So I will say, most of the work that's been done on this parasite, up until now has really been heavily focused on the immune mechanism. So it's actually pretty amazing. Like, we thought from sort of 1999 to 2008, we had a really good sense of how C. elegans combated its pathogens, so what the immunological pathways were. And then folks discovered this natural parasite, and it sort of totally changed the way we think about immunity, right? Because it's actually tapping into all sorts of different pathways that were previously just sort of unknown genes, unknown pathways. And it's pretty well characterized. So the immune system the immune response is well characterized. I think one of our goals is to come in and say, okay, based upon what we know about the ecology and the natural history of these organisms, what are the actual defense mechanisms they're tapping into, right? What are they actually using? What can evolve most readily? And so that's where the dispersal angle has come from. But that last bit argument that I was making was that they may trade off with one another, right? Not potentially because they're both costly. We do think there's evidence that immune responses are costly. We think dispersal is costly, so you could imagine there'd be trade-offs in that sense. But they could also it could just be that dispersal just prevents the evolution of downstream defenses, right? If that makes sense, right? If you're dispersing really early out of an epidemic, you're very rarely encountering parasites much less frequently than if you're a genotype that doesn't readily disperse, right? So you could actually, just by dispersing early, you could render these sort of downstream defenses, these physiological defenses, selectively neutral. So those are kind of competing ideas that we're interested in. And we can get at those in a couple ways, right? You can just look across these large panels of strains we have and say, okay, what's your dispersal strategy? And how resistant are you when we force you to interact with the parasites? And are those negatively or positively or not correlated at all? I mean, alternately, we can also experimentally evolve it, right? We can sort of force different dispersal strategies and see how resistance evolves under those different conditions.
Thank you.
Does that answer
Very
your question?
interesting.
Yeah. Okay.
Thank you, Luis.
So, two short part questions.
Sure.
In nature, when these nematodes get dispersed into new bacterial blooms, how likely is it that those are pristine and sort of unoccupied by other nematodes, if you have a sense?
I don't know. I think the working assumption is that they're pristine.
Mm-hmm.
It's probably not that nice.
Yeah.
Yeah, I mean, this is one thing that's motivating some of that field work, right? We don't know things like just basic occupancy of the nematodes. We don't know incidence of the parasite, and both of those things would kind of predict. But yeah, no, we operate under the assumption that they tend to colonize new patches, right? That are pristine, and that's somewhat supported by the genetic data, and that frequently it's a single selfing lineage. Mm-hmm. But certainly they're arriving sometimes. They're going to be potentially frequently arriving into patches where there might already be nematodes, there might already be infection. I'm sure that can happen. We don't know the rates at which it happens.
That changes how important my second part is.
It would change the way we're thinking about this, for sure. Yeah.
Do you think it's possible that this is a parasite-adapted trait? So increasing dispersal of the host, bringing new pathogens into these patches with susceptible hosts?
Yeah, I don't think we can rule it out. I think I thought it was more possible before we started getting some of this more recent data, which is supporting. Well, maybe they're not mutually exclusive. I have to think about that. Yeah, I do think it's possible because. So one thing we don't know. We look at just that colonization stage, right? We just say we're going to do dispersal for you. And then can you reproduce? And when we do that, we do know that some of those individuals that arrive can still spread, right? They can transmit parasites even though they can't reproduce, which that would support this model that it could be a parasite adaptation for transmission. But we haven't quantified it yet because we're just like working out some specifics of the experiment. But I'm pretty sure a lot of these dispersers, they're not just stuck at the colonization stage. They also have some other issues that would affect the transit stage. Like, we're pretty sure they're much less desiccation resistant and not as long lived. And that would take them out before they even get to a new patch. So that's the other reason I kind of lean away from the parasite manipulation hypothesis, but I can't rule it out. Yeah, but that's why those experiments that I keep kind of putting off are important.
Oh, you covered.
Let me go back. Sorry.
Oh, you just covered, yeah.
Was there another one?
Questions everywhere.
That was a fantastic talk. Thank you. I am just having trouble wrapping my head around that result that they all die when you move them after they've been exposed, but then a bunch don't when you move them a day before that.
Yeah, that was also very surprising.
And I guess my question is, like, why? Yeah, I don't know. I mean, why is kind of the shorthand, but like, why are they? You're doing the hard work of transporting them, and so you're just putting them in a novel environment that has lots of resources. Why are they all dying?
Yeah.
Compared to the population where they've been exposed, but you're not moving them? Like, what about moving them kills all of them?
Right. Well, okay. Yes, good questions. Couple thoughts. So first of all, they don't. I wouldn't say in this I mean, these are pretty exposed populations by the time, and these are like spores are really building up. So it's not like things are like just super peachy over here. Like these the ones that stay are also doing poorly. They tend to get out some babies and to not be sterilized. These individuals, they don't actually die. They eventually die. But the main thing that's happening is they cannot mature reproductively.
But so if those dispersing individuals are like random draws from the population that's been exposed at that rate, then shouldn't that population's Malthusian fitness just crash? Like, shouldn't it be done?
Oh, yeah, they're definitely crashing.
Oh,
yeah.
okay. So this is like a life raft from a sinking ship that also sinks.
Yeah, exactly. Right. Yes, that's nicely put.
Very curious.
Yes, right. Which is why getting out early is really your only solution.
I'm on
right.
board.
Yeah. No, these populations are crashing. Why only like a day earlier? I'll be honest, I actually kept forgetting that Lewis only did this a day earlier, and I kept being like, why did you do that? But now it makes sense, because now that we have the prevalence data, which I'm not showing you, but we know that, I'm trying to respect that Lewis doesn't want me to show it yet, but I'm going to talk about it, which is that it is really, really low. It takes a long time. It kind of is starting to tick up, and then it just booms. And actually that prevalence changes over the course of a day in the context of these Petri dishes. So, it's contained. So it is this really dramatic change in your infection risk. So that's why that result that didn't really make sense to me, now that we've tracked prevalence through time, makes a little bit more sense. Yeah.
Cool.
Yeah, that's a good question.
I think Gina looks like she really, really has a
I think I was wondering, is there any selection, is the early dispersal strategy, does that lead to any selection on the parasite itself? And because I was wondering if there's just like a mismatch, and that explains your data.
Yeah, I think the only reason this is potentially working is that our parasites actually have fairly long generation times. So the host generation time is 3 to 4 days. The parasite generation time is about 3 days. So it's about, it takes about, here's where we think like the first round of transmission is happening, or like right in here, because it takes about 3 days for spores to get pumped back out into the environment, and then another day for sort of infection to really set in. Right, so this could work it's only when sort of that dispersal can happen before infection risk really starts to take off. So there's some assumption here that infection risk is not just increasing incredibly rapidly. There's a window of time before your risk is really high. And I think that's more likely to be happening in the natural environments. We haven't done this, but this is just like an open petri dish. Parasites are just accumulating, they're sitting around, but in the natural environments you have a lot of structure. So I think the potential for this to happen, to just be around low parasite prevalence for a while, even as you're sort of in an active epidemic, I think that's more likely in natural settings, which we could probably start testing. Does that answer your question? So, yeah, okay.
I had one question. Earlier, like towards the beginning, you showed the video of them like having that behavior of dispersal. Does your lab like look at changes in behavior at all with infection? Like, do you see an increase in that dispersal behavior when you introduce this parasite?
I'll just put it on so we can look at it.
No, I really don't like it.
You don't like it? Okay, we'll put it on. I love it. I'm sorry. It's so cool. Yeah, so that is actually related to Louise's question that I was kind of alluding to. They have all these experiments that are planned that we haven't quite got around to, of looking at, in particular, that nictation behavior. So we think that they are less able, we think that they're less able to nictate, which is that waving behavior. We also think they're more likely to desiccate, but I'm really interested in basically testing, right? Put X number of dispersers on a plate and count the number that can nictate. We have to modify our plates so they can actually, you don't typically, the reason people don't know about this behavior that much is because the dowers actually get stuck with the surface tension to the petri, so they can't nictate. The only way they can nictate is they find like a fungal contaminant that they can climb up and wave, or if they find the dried edge of plates and they can go up. So we'll simulate by putting medical gauze on the plates, and they'll climb up the medical gauze. But we're just trying to figure out a controlled way of measuring those things. Yeah.
That's really cool.
Sorry, I'm asking another question.
Sure.
So building off that, the dictating the dower behavior is so interesting. I guess I'm curious whether, because the dower is also this desiccation resistant, long-lived life stage, if it's also a mechanism of diapause, of escape in time rather than necessarily escape in space.
Yeah, that's a great question. And certainly that's how we viewed the dower phase, because it was only ever worked on in the lab, and basically researchers would let their plates dry out, and then they'd come back and they'd be like, oh, it's okay, they diapaused as dower, we can save our population. So it was used as a diapause stage. And it could well be. The nictation, that behavior argues that it's for dispersal. To what extent can an individual, they are very long-lived. That is totally true. I mean, in the lab, I don't know in the field, in the lab you can go from something that lives maybe like 5 days to something that can, well, in the lab it can live for 20 days, but to something that can live for 4 to 6 months. So definitely that's consistent with diapause. I have a hard time it seems so unlikely that a patch gets reinvigorated, right, or whatever, or renewed in the wild. Like, these are such ephemeral, they collapse. Like, it's hard for me to sort of grasp like what the benefit of sitting and waiting would be. So that's why I tend to think of them as dispersal stage, and we see them on vectors. But it is, it's a good question. That it could be both, right? It could function as both. Kind of similar, like Daphnia Phippia are both sort of they disperse in space and in time. Yeah.
Great. There's 2 more people who really want to ask questions. We're going to do them, and then we'll give you a break.
Well, this might be a little bit of a change of subject, considering everybody was so interested in dispersal. Have you considered the possibility that hermaphrodite sulfate might be a sort of a middle road to mediate the costs of sex and the costs of parasite risk?
I think about this all the time. So we have started to look at this a little bit in the lab, and it's actually one reason I sort of got into this is that we know, right, C. elegans, it's androdioecious. You have hermaphrodites that can self-fertilize, they can also produce males, and you can outcross. And we know that in lab conditions, you can simulate coevolution with parasites, and it can drive male frequency up so that it functionally becomes an outcrossing system, right? So absolutely you can see the red queen alive and well, but then you go out in the wild, and the lore for C. elegans is that it's selfing 99.9% of the time. And yet it still maintains males. So that has bothered me, and it's one of the reasons I wanted to transition into these natural parasite systems to understand like what might be different in the wild to what we see in the lab, where sex can be maintained. So we've started to look at some of these wild strains, and I won't show these data, but my postdoc Liao has shown that these wild strains actually vary enormously in their male production. So some of them don't make males at all, some of them can make 50% males. So there's a lot of variation in selfing versus outcrossing in this system. Parasites seem to cue male production. So yes, I think there's something there. And part of, like, I think you kind of saw this setup, a part of this dispersal angle is trying to understand, well, can parasites co-evolve with hosts in these systems, or are they dispersing so much that you can't actually see the red queen operate? So and that's alleviating selection, or that's reducing selection for sex. Yeah, that's kind of probably more than you asked, but things that I think about.
Last question in the back.
I'm going to shift the question focus more towards the first part, to the snails. And like I know that oftentimes snails are the intermediate hosts for nematodes and trematodes, and that like for the New Zealand mud snails, there are multiple different parasites like trematodes, like microphallus has like crab as their final host, and then you have other fish as the final host. So I wonder if you have any data on like the infection rates in the final host and whether that affects the infection rate and triggers any kind of like differences in the sex, whether it's sexual or asexual in the mud snails. And also, as final hosts usually are sort of like the predators for the snails sometimes, and so whether like the predation cue can be something that is also a factor that affects the sex, the reproductive method in those mud snails.
Yeah, so I'll say to most of those questions, I don't know the answer, but so actually this is microphallus. It's been renamed.
Okay.
So yes, and in this case, the definitive host is actually dabbling ducks. It is very similar to schistosmiasis, where it's just moving from snail to duck and back. And right, so it is predation. So the duck is eating the snail, and then it's releasing eggs of the trematode back into the water column that the snails are picking up. Yeah, so I didn't talk about that at all. We don't know how that affects many aspects of this system. There are some things we do know, like we do know that these snails will kind of there seems to be some manipulation of the snails to sort of put themselves in locations where they're likely to get eaten by ducks when they're infected. We also think the distribution of infection around the lake is heavily driven by where the ducks are. So one thing I didn't say, there's an enormous amount of variation among these sites. So part of my PhD was really like sampling sites all along the lake and trying to understand why we see such different infection prevalence, and we think it's driven by the distribution of the definitive hosts. In particular, kind of like hanging out over here and getting really high infection rates. And that can like churn coevolution more quickly. So we have what we think are coevolutionary hot spots in certain parts of the lake and coevolutionary cold spots. And I kind of didn't talk about the definitive host, but certainly is critical. We also think it's critical, right, for the parasites. So that's where the parasite's having sex, and the parasites got to be maintaining an enormous amount of genetic variation to be able to fuel these coevolutionary cycles. So yeah, good questions. And there's lots of other trematodes that I didn't talk about. We think, at least historically, it's Atriophalophorus that's been the most abundant, and that seems to be what drives the coevolutionary dynamics. But there's lots of other cool parasites.
Very cool. Thank you.
All right, let's all thank Mandy.